So hello again in English. So the title of my talk is From Biomolecule in 3D to Collaborative Research in Biology. And my name is Philippe Youkharibache from the National Cancer Institute. And it's a collaboration with Jiao Wang from the NCBI structure group. So we've seen so many talks about AlphaFold that it's clear that sequence structure function paradigm has become central now. And it's going to be interesting to see how it plays out. I'm going to give a little background on where we're coming from. So interactive molecular graphics has been in the works for about 60 years now. And in hindsight, you can understand that it's been an interplay between different technologies that emerged and converged. And finally, where we are today is because of that. iCn3D is actually a web-based platform for visualization, analysis, and sharing of protein models. And the question is, what does the web bring that we didn't have before? It's actually collaborative research capabilities so that we have interactivity not just with a protein model, but between scientists. So it's important to understand that interactive molecular graphics and protein folding are two faces of the same coin. And it all started with Cy Leventhal in the 60s, where he was doing molecular model building to observe giant molecules on a screen and try to understand how actually they fold into the shapes that nature gives them. Now, fast forward 20 years, in the 80s, I was privileged to join Cy Leventhal at Columbia University, thanks to Barry Honig, also at Columbia. And this picture is interesting to me, first of all, because that's the only one I have with him, but also because it's a point in time and space. That's the NIH for a meeting in '88. And I didn't realize at the time, I had moved already into the industry to develop software for molecular modeling precisely. And I didn't realize that I would join back the NIH much later in the about 2014 to basically finish the job. So I think it's important to realize that there have been a lot of development since the 80s, in particular in the graphics part with Silicon Graphics and the development of the graphics library that led to OpenGL and much later, after JavaScript, to WebGL, which is what allowed us to develop iCn3D much later. So, of course, everybody knows the World Wide Web emerged at the end of the 80s, but it's only in 2015 that we were able to start developing iCn3D as an open source web molecular graphics software, first for visualization, then for analysis, and later on, especially when the pandemic hit, for collaborative web research. And now we're working, of course, on much larger data set since COVID-19, but now with AlphaFold even more. And the interesting thing is that there is an interplay between these 3D graphics and protein folding efforts, especially originally the protein folding was more like coming out of the force field world with molecular dynamics, and in particular CHARM. And these programs like CHARM merge and integrated with molecular graphics with commercial companies like Polygen and Biosim. I participated to both. And later on, in fact, the GPUs coming out in play also, provided a platform, a computational platform for machine learning. So there is this interplay between the two, but the very important part is that PDB in 1971 was open data. And that open data allowed, the development we know from AlphaFold. Not on just open data from PDB, but the effort led by John Moores at the time with CAS starting in 94, leading to what we know now in Cas14 in AlphaFold. So what is important, I think, in this picture is the amount of technologies that integrated and intertwined, and the fact that some developments were possible only because of open source and open data. And now we are reaching the point where AlphaFold, RosettaFold, ESMFold, and FoldSeq are big players in the game. But the question is, how do we manage the data, especially between scientists? So in terms of software, web molecular graphics is basically doing what you do with classical platforms, mostly commercial. There's no such thing as open source in this field, unlike genomics, for example. So on your right, it's kind of a parallel between the current commercial software and the web software. But the important thing is, how do you share data? How do you share data? And more than that, how do you share analysis? How do you share molecular scenes? This is something like has never been possible before, sharing scenes, unless you have the same platform, computational platform. So in a nutshell, iCn3D has 3 dimensions, if you want, from a user perspective: interactive visualization, interactive analysis, with a lot of features that you find somewhere else, but also you can transform that interactive analysis in batch analysis thanks to Node.js. And the third part, which is the one that I want to insist on, is a collaborative and sharing part, where you not only can share the live molecular scenes, but the data that allowed you to create these scenes, the analysis that led to these scenes, and the software that allowed these analyses all into one link. So, everybody knows probably in this audience the spike protein, given the fact that we've been all affected by it. And here you see a visualization. In the middle, you can combine different PDB to understand better an environment. Here with the ACE2 protein receptor binding the RBD in the up position from the spike, and then a blow up zooming in on the interface. Now, at this stage, I would do an interactive, because it's interactive, demo, but actually, I will run the movie that I did a couple of days ago. How does a movie run?
Movie?
It's here. Normally, I should click on that.
It's an X? This?
It's in the presentation. No, not here. No, it's a previous.
Previous? Okay.
Yeah.
Okay. This?
No?
This?
No, normally this, I'm sorry, go ahead, go back one, here. No.
Your project.
This one.
This one?
Is a video.
Okay, let's go.
Yeah, I'm sorry. The link is expressed here. We're playing all the commands that were used. This is my voice. Including the date of the analysis, so that 10 years from now, if the software changes, and it will change, the right version of the software is used to reproduce the analysis. So we deal with the reproducibility. At the same time, here the 2D becomes 3D, with all the coloring here for all the different parts annotated and all the NCBI annotation available. When zooming in on the interface between the ACE2 protein here in gray and gold versus the RBD in blue and red, especially these blue and red meaning conservation, red being conserved. We can compare, for example, the SARS-CoV-1 versus SARS-CoV-2. So SARS-CoV-2 here, all the wiring done and rewiring between SARS-CoV-1 and SARS-CoV-2, the software being able to pick up the interactions that were conserved versus the new and old interactions in the SARS-CoV-2, where you see a lot more hydrogen bonding between side chains of the RBM with the ACE2 interface. So while the two look very much the same when they're superimposed, there is a change in the wiring, and definitely it seems like the SARS-CoV-2 binds much more strongly to the ACE2 receptor. Now, the strength of iCn3D is to be able to annotate from a user perspective the different regions of the proteins, and one can actually zoom in here. The interface, of course, the interface is picked up automatically by the software, and one can visualize the differences here as they are also shown in the networks of interactions. A lot more analysis have been done when this pandemic occurred, when we have a preprint available out there that you can consult on bioRxiv, and lots of different links representing different analyses of different viruses. At the time, we didn't have all the variants, but the same analysis can be done for variants of concerns. But here, for example, we can compare MERS versus SARS and so forth. And the software is available on GitHub. It's open source, and lots of tutorials are available on the web, not only by us, but others. Here, a gallery of examples that you can look into. And this is available for you to consult the different capabilities of the software, for example, what we just looked at. In teaching, of course, they've all understood how they can annotate, structures they want to explain, enzymes, membrane proteins, and so forth. So if you click on this guy here, lipids, you might end up looking at, for example, this example, and this is embedded into a web page of a course, and all the commands of the software are available here. For example, you can view or color or select different parts, like for example here, colored by spectrum for chains. These can be also embedded into Jupyter notebooks in the same way. Here you can invoke any command or any URL that you want from iCn3D by just invoking first iCn3D within the notebook, and same thing, the same interface, which is actually the interface you have on your cell phone. In publications, here we published a paper, for example, recently, where for cartoons you can invoke directly a link instead of having the supplement and going and fetch a supplement, finding the software, loading the PDF file, and so forth. Just click here on this particular link in a repository. We've put the file, the model, and the software is available to analyze the structure itself. This iCn3D has been used also in you know, web services for databases, like here is the COVID database with all the antibodies that are valuable today for against SARS-CoV-2, and they're using the viewer there where you can show where the epitopes are. Oh. Yeah. So I can go to the interactive one. So I think I showed you a few examples of applications in research, in using repositories. By the way, everything is fair to me natively, but we can discuss that. In education, of course, they are probably the biggest users right now because it's free, but also because they can exchange with their students assignments and courses and textbooks. The embedding in Jupyter has probably a lot of potential. The embedding into databases and web services. And in publishing, unfortunately, publishers don't embed the applications. You have seen that it's just a link that has to be visualized as a link. But hopefully in the future it will be available. I didn't show you the demo, and I won't do it because it's interactive. But nowadays, you can simply paste a sequence here and invoke a server like ESM and interactively in 10 seconds, this will fold. So this is like the dream I would have liked 35 years ago. And you can change the sequence, compare the sequence, align them, figuring out what your impact is interactively. You don't have to run and wait for machine learning to tell you what to do. So this is about, and today what you see on the right here is a dimer with a nice cysteine bridge at the hinge level of CD28 you get from AlphaFold or from ESMFold. Contrary to what you heard, you can get good dimerization with ESMFold as well. So of course, the next step is about getting from structure to function, as we've always believed we should, and a higher level of complexity in quaternary complexes. So I'd like to mention what was SAI's philosophy. SAI's vision was about using interactive molecular graphics to build, visualize, and analyze 3D models to rationalize experimental results and get ideas to guide experiments, exploring molecular and cellular mechanism, and cycle through this process to improve both models and our understanding of biology in molecular terms. And today, iCn3D helps in collaborative process between modelers and wet lab scientists. In fact, I use it a lot to exchange with wet lab scientists, even MDs, because MDs actually design proteins for CAR T-cell therapies without us, and we're trying to rationalize what they do, and hopefully we will help them for the next steps. So tomorrow is wide open. I think it's open data that's important to understand. For example, PDB got us to AlphaFold, and the next problem is how do you actually manage to collaborate between scientists? What kind of software can you use? And I think iCn3D is positioned there. And to finally, I will thank some collaborators, at NCBI, J.R. Wang in particular, Tom Madesh, James Song, Aaron Marshall-Bauer, and Steve Bryant, who gave the go to the project. Of course, Tom Mistelli, that gave me freedom to operate, to be able to get where we are today. And of course, the early open source collaborators outside of the NIH in particular, Ravi Abrol, Raoul Cachot, David Bell, Alisa Dillman, also Jianlin Cheng, before AlphaFold, where we were working on machine learning for designing dimers. And the WPDB guys like Samir Velankar, Alexander Rose, Peter Rose, and special thanks, of course, to Diane Kovacs and Stephen Lurd and Seth Mulholland at ISMB because we've been running hackathons that helps a lot in developing iCn3D. And of course, my former mentors in the past, Barry Honig, the late Cy Leventhal, Arnie Hagler, and not to mention also Chris Henn in Inside Reality, who 20 years ago was developing immersive virtual reality. And today we can get that at low cost. And I think we're going to have development to that area together. With that, I would thank you for your attention and take any question.
I think we don't even have time for questions, but if there is one urgent question, if not, you can talk with Philippe outside. No questions?
One
question. Well, that's great. At least one.
I'm new in the field, so I'm asked a very simple question. So if a student want to use this software, do they have to download, install, or I can install on as a web server that they can use a browser to log in and then to play all those wonderful things?
I'm sorry, can you repeat the question?
Oh, okay, so basically it's the installation. So if my student want to use this program, do they have to download, install on their computer, or I can install on a web server, then they can use a browser to log in and then to play with
Very good question. It's a completely open source. You can download it, install it in 5 minutes on your own computer, and have it on an intranet. You can have that. You can have your file served with a web server. It can be completely autonomous.